forgot to tell you that he has been awarded with Dara Imtiaz and Hilali Imtiaz by Government of Pakistan. Please give a big hand to Professor Dr. Shahid Jain. Next to him is sitting Dr. Abdul Haq, who was a deputy chief scientist at NIPG and pioneer in vaccine research in National Institute of Biotechnology and Genetic Engineering, Faisalabad. He was the ex-vice chancellor of University of Faisalabad and currently serving as director of World First. Please give a big hand to our guest, Professor Dr. Abdul Haq. Sir, mic is yours now.
Bismillahirrahmanirrahim. Thank you very much. Welcome to the first technical session of this conference. And in this session, we have 5 speakers. The first speaker is Professor Michael Hess. Is he around?
Yes, he is.
All right. He's from University of Veterinary Medicine, Austria, with a distinguished career spanning over 3 decades. Professor Hess is widely recognized as a leading authority in poultry diseases and virology. Professor Hess' contribution to the field are exemplified by his pivotal research on avian adenoviruses and his leadership roles at esteemed institutions such as the University of Veterinary Medicine, Vienna, recognized for his outstanding achievements. Professor Hess has been honored with prestigious awards, including the Carl Friedrich. Did I pronounce it?
Yeah, I think so.
Okay.
That's pretty
Correctly?
good.
Okay. Research Award and Robert Fraser Gordon Memorial Medal. We eagerly anticipate Professor Hess enlightening discourse, which promises to enrich our understanding of vaccination strategy in poultry health. Professor Hess is to deliver a plenary talk on vaccination strategies for selected poultry pathogens. So it's over to Professor Hess.
Yes, thanks a lot, everybody. Good afternoon. First of all, I want to thank the organizers, especially Professor Rahman, for inviting me here to come to Pakistan again. A country I've been there before. I like the hospitality and the people here very much. So it's a real pleasure to be back. Thanks for the chairman for the kind introduction. And now for the next 30 minutes, it's up to me to keep you alive after lunch. Knowing where your blood is circulating, actually, I try to motivate you. Vaccination strategies for poultry pathogens. I will talk about 3 pathogens. I cover this field and cover the research area which we're covering in the last, let's say, 10-15 years in those research areas. And I will kick off with a parasite, which the majority of you don't know, but I think it's an important subject for a vaccination conference, and I will show you why. And then I will go to Escherichia coli, an important bacteria, and of course, as the teacher mentioned, I cannot give a talk without talking on adenoviruses. Let's start with the parasite. The parasite I'm talking about, Histomonas meleagritis, as I say, it might be unknown for you. It was first described in 1893 as a devastating disease in turkeys in the US. And then there came a research period where a lot of effective drugs were detected, especially imidazoles, and they are highly efficacious. At the same time, people started already to work on immunological data, on efficacy studies to see if birds can be vaccinated. However, between 1976 and 2003, there's only 1 research paper in this area, 1 research paper. And why is pretty easy to answer, because there were very efficacious drugs and there was no research needed. But this changed. This changed in Europe and this changed in the US. The FDA and the European medical agencies have banned all therapeutic and prophylactic substances to treat and to cure this disease. And the headlines when this ban came into play was blackhead, which is another term for the disease. Blackhead may kill the European turkey industry, which it hasn't done. But there are some farmers who have given up in the meantime. And it's a serious animal health and welfare concern. Just to give you some data, there are not many data because it's a non-notifiable disease, so you are not obliged to notify the disease to the authorities. But you can see from those data, in the US we have about 100 outbreaks per year in turkeys. and in Canada. And you can see from this survey, you can have mortalities up to 60–70%. On the right, on this chart, you see an outbreak in turkeys in Austria. These are 7,250 turkey toms, and you can see mortality rates up to 900 dead birds per day. So at the end of the day, you have nearly 100% mortality. It's a very cruel disease. And this is the pathology. If you miss anything in your life in pathology, you'll never miss blackhead if you see it, because the lesions are so prominent in the liver, in the ceca, as you can see here, but also in chickens. Nowadays we see it also in chickens. And we see it a lot with colibacillosis in chickens, which forces us to use antimicrobials, but we are forced not to use antimicrobials anymore. Now talking about vaccination, and I will drive you through the road we have taken in the last now nearly 20 years. Tissa, he reported already that it's possible if you isolate the parasite and you passage it in weekly intervals, it might be attenuated. An important study comes from Clarkson. Clarkson did something on passive immunity. He infected birds, he treated them that they survived the disease, he took the sera and took the sera to susceptible birds, and he showed they are not protected. So what does that mean? Antibodies are non-protective. You need a live vaccine. And this is the great disadvantage, because the pathogen flagellate is very unstable outside the host. It has a low tenacity. It survives in the environment only for 1 day. And in nature, it survives in the vector, in the nematode oocyst. Now, Lund questioned the concept of in vitro attenuation. He said it's never possible to attenuate this parasite. When people claim they attenuated, they have simply isolated it, but in that isolate was also an attenuated strain, and when they make serial passages, they simply outweigh the virulent one, and they select for the attenuated one. And this is something we wanted to disprove. And this was the start of our exercise on vaccination. Here you see an isolation, and what you easily can see is these are flagellates, these are moving parasites, and you can see they have different forms, they look differently. So the first question is this a relative of this one? Is this a relative of this one? Etc., etc. This is all what you don't know when you do this, because they can change the morphology. And we established the concept of clonal cultures. These are clonal cultures. What we did is we take a single cell, and those cells, they replicate by single cell division. So from a single cell, you can establish a clonal culture. And that's what we did. And we named those cultures. The terminology is simply we copy-paste from influenza viruses. Histomonas meleagritis, Turkey, Austria, 2922, clone 6 from year 2004. So pretty simple, we just did copy-paste. But now we have clonal cultures, which means we can go back to a single cell and propagate the whole cell. And we passage the cell for 3 years. And we put it back into turkeys, and the turkeys survived. And we challenged the turkeys, and the turkeys survived. And you can see the videos on the vaccination trial. These are the non-vaccinated turkeys, obviously already 3 days earlier, because they all going to die. As I say, it's a very cruel disease, as you can see here. And these are the vaccinated turkeys. I think the difference is pretty obvious what you see. We looked into cross protection, isolates from different countries in Europe, Austria, Germany, France, and so on. The green ones are the vaccinated ones, and the red ones are the non-vaccinated ones. What happens with the vaccine? The vaccine itself resides mainly in the cecum, as you can see here, by immunohistochemistry, and you don't find it in internal organs. This is completely different to the virulent parasite. We also did a trial on back passage, reversion to virulence is an important aspect in live vaccination, as you can imagine. And we did this 5 times in turkeys and chickens, and we did it then in a cohort of 30 animals, and you can compare the virulent with the attenuated one, and then you see certain lesions in the ceca, but not in internal organs. But you have another problem. The parasite cannot grow without any bacteria, any live bacteria. He needs live bacteria. And we don't know why. We don't understand why. But this is known for 60, 70 years. You cannot infect monobiotic chickens or turkeys. The infection will never work. So for example, this is the parasite. This is a picture, electron microscopy of the parasitic cell, and this is the bacteria. The parasite takes the bacteria up. And this is a great disadvantage in establishing a vaccine, because you have a defined parasite, but you have a mixed microbiota culture. And in order to overcome this, we established a concept where we worked with a GFP-labeled Escherichia coli, and we exchanged the microbiota with this GFP Escherichia coli. In red you see the parasite, and in green you see the Escherichia coli as it's taken up. And by this we were able to establish a shuttle system to knock down the GFP-labeled E. coli and to substitute with another bacteria. We also worked a lot on immunology. What is the difference between chickens? What is the difference between turkeys? We see a difference in the early pro-inflammatory response and a higher presence of especially interferon gamma in chickens in comparison to turkeys. And we also looked what is the vaccine doing in regard to B cells, circulating B cells, but especially in CD4 and CD8 T cells in the cecum. Now, after the parasite, I switch over to the next subject, to the next selected poultry pathogen, and this is Escherichia coli, and this goes closer to what you see in the field, because everybody who is in the poultry field, I think colibacillosis you must have seen, otherwise you are not involved in poultry diseases, I would say. So, this is just a scratch from the pathology table. As you can see, Escherichia coli infections. You may have perihepatitis, you might have pericarditis, of course, you might have hepatitis, you might have salpingitis, ophiritis in older birds, or you might have yolk sac infection. So we have huge panels of infection. The great problem with Escherichia coli, and Escherichia coli is especially evident, is the great heterogeneity of the isolates. So the isolates hardly match the genetic type. The phylogroup hardly matches with the pathotype. In pathotype, we usually differentiate between AFEC, avian fecal E. coli, or APEC, avian pathogenic E. coli, as we have published here, a survey on nearly 2,209 full genomes. And what is especially of importance, and I want to highlight this for people in the field, is a long story which I make very short with this picture. This is a typing, PFGE, pulse-free gel electrophoresis, typing of Escherichia coli isolates. And what we have done, what nobody ever did before, is we took isolates from the same organ and we typed them from the same organ, and you get different isolates from the same organ. And this is also phenotypically the case. They have a different antimicrobial resistance profile. So for your treatment concept, it depends heavily, maybe, or it is influenced at least from which strain you're taking for your analysis. But anyhow, despite the fact that Escherichia coli is such an important disease, there is not much known in vivo pathogenesis. How does the disease goes in vivo? And that's what we are interested in. But you have a problem. Every chicken carries Escherichia coli. So how do you know in an experimental model that the strain which you're putting in is the strain you're getting out? Because they are full of E. coli, every chicken. And for this, we established the concept of bioluminescence imaging. We label our Escherichia coli genetically with a bioluminescence gene, which is self-reflecting, and then we do a simple animal experiment where we infect chickens to learn about the disease, the high mortality, which you can see here. But then you can use the carcasses and you can trace the bacteria more or less where it is, at which time point, as you can see it here in the full-blown colibacillosis, and then later it resides in joints in the air sac, for example, and in individual organs, as you can see here in the ovary and in the oviduct. So the ovary and the oviduct are obviously a certain target organ of Escherichia coli as well. And that raises the important question of vertical transmission for the day of broilers, which is indirectly confirmed by genetic analysis so far, but it might have a consumer impact on table eggs if you spread the bacteria as well via the ovary and the oviduct. And for this we did extensive immunohistochemistry, but we did double immunofluorescence for Escherichia coli on one side. This is the green, this is the bacteria which you see here. This is Escherichia coli. And we did a labeling for albumin as well in the magnum. And the albumin is what you see here in red. And if you merge the pictures, then you can see the Escherichia coli is not only in the ovary, it's also in the secreted products of the oviduct. So we find it in the yolk, and we find it in the albumin as well. But let's go to vaccination. Our approach to vaccination was, first of all, we did an infection trial where we infected young birds by different routes. We wanted to understand the disease again, what is happening in the bird. And we used aerosol, intranasal and intratracheal infection. And aerosol infection we do in isolators, as you can see here in a model. This is the isolator. We have a plastic box and we have a nebulizer outside, and with this we infect the birds by aerosol. And for vaccination we applied the concept of irradiated bacteria. Irradiation is a concept which is widely used for parasites for a long time, and recently more in now joining also the field of vaccine development in virology and in bacteriology. We used irradiation to create what is called metabolically active but non-replicating bacteria. Metabolically active, metabolic activity is measured by the Alama Blue assay. It's a proliferation assay, and you can see for a long time the metabolic activity stays, and it differs between strains depending which strain you're using. We looked on electron microscopy on those bacteria, how they look like, and you can see this is the untreated bacteria and this is the treated irradiated bacteria. You have the fimbria nicely present on the surface of the bacteria. This is of course an idea of this concept, to have the fimbria present, because when you add, for example, when you do formalin inactivation and so on, the fimbria are lost, of course, in the production process. If you measure the proteins by multitoph MS, then you can see on the top three, the blue bars, those are formalin inactivated bacteria. You see only a few peaks, and these are the irradiated bacteria and the live bacteria in black and in red. They look pretty much similar. And then we looked, are those bacteria which we irradiate, do they induce immunity in vitro, in an in vitro system? We take spleen cells, we take blood cells, and we take lung cells, and we compare live bacteria, for example, with irradiated bacteria and killed bacteria. And then you can see at various stages the irradiated bacteria next to the live bacteria in inducing a certain expression of cytokines. And then we did the first efficacy study where we vaccinated birds at day old, we challenged at day 14, and then we did necropsy, and we used 2 different challenge strains also to look a bit into heterogeneity of the protection. And you can see the moderate clinical signs only in non-vaccinated birds. I do this in brief. I mentioned the publication here for those who are interested in where we have published this. And if you do histology and if you apply statistical analysis, then you can see the probability for lesions in non-vaccinated birds, which is characterized by the right bars, is much higher in comparison to the vaccinated birds. And if we look to immunology again, then surprisingly, of course, in re-stimulated cells, to highlight this in the lung, for example, you can see for CD8 alpha positive cells or TCR gamma delta T cells, you have a high level of expression in vaccinated birds. So this type of vaccine is inducing, of course, a certain immune response, which is important, might be important in production. This needs to be further detailed analysis, of course. Now the third pathogen I want to tackle is adenoviruses, as I mentioned before. In the last, or put the other way around, start at 1963. 1963, Hempel and Fraser described in the US inclusion bodies in the liver of a chicken, but with unknown etiology. This was the first report. And these are case reports on inclusion body hepatitis. What you can easily see is that the number of case reports in the last 15 years went up. Case reports are not easy to publish sometimes nowadays, because the journal says, oh, we know about this, because it's happening now in Morocco or wheresoever. This is not of interest for our readership, this is not new, this message. Publish it somewhere else. But you can easily see how the numbers of publications went up for IBH. And in 1987, HHS, hepatitis hydropericardium, joined the scenery, as you can see here, with this bar as well. And then adenoviral gizzard erosion came up as the black line as a new disease. Nowadays we can say that IBH is globally spread. There are globally lots of vaccine used, lots of autogenous vaccines are used globally, in North America, for example, in South America, but also in Asia, of course, in Australia, live vaccine is used. HHS, hepatitis hydropericardium syndrome, first appeared 1987 in Karachi Ghost, in Angara near Karachi Ghost. That's why in Pakistan the disease very often is called Angara disease. The Indians call the disease lychee disease because the heart has a type of, like the lychee fruit, more example, that's how it looks like. So what is the difference between HHS and IBH? The difference is simply the mortality. In HHS you have a much higher mortality reported in the field as well, whereas in IBH you have a lower mortality. These are pictures from experimental studies in SPF birds. What you're seeing here is SPF birds infected. So these are not birds from the field which might have a co-infection with infectious bursal disease or CAV, immunosuppressive whatsoever. This is not the case. And what you can see is, of course, the hydropericardium. You see the hepatitis nicely, but you can see you can have also bleedings in the thigh muscles. We saw that, depending on the strain which we use. You see bleedings in the thymus, in the pancreas, we see a nephritis, and we see an atrophy of the bursa. For a long time it was thought that foul adenoviruses need immunosuppression. If there's no immunosuppression, you have no disease. But this is not the case anymore. I think this is pretty clear now, that we have also outbreaks where immunosuppressions might contribute, of course, they contribute, but they are not the sole cause. And why is this? Why is this, and why did the number of cases went up in the last 15 years? There are two explanations which we actually have. One explanation is, if you look at these two types of birds, one is a layer and one is a broiler. It's easy to recognize. All experimental studies 30, 40 years ago and even today are done in SPF layers. But IBH and HHS are metabolic diseases. And these two birds have a completely different metabolism. Completely different. One is reared for growth, the other one is reared not to grow, more or less, not to put weight on. And we use the same viruses and we put them in layers and broilers, and indeed the outcome was completely different. Either if you use broilers with high mortality or if you use layers. And why is this? The broiler runs in the diabetes mellitus because the pancreas is also infected, and the broiler has much more difficulty to overcome this. And that's why in the field, I'm pretty sure you see much more outbreaks in broilers than you see in layers. in layer type chickens, put it that way. So this is one explanation. So in fact, in a certain way, we breed birds for disease susceptibility. And the other theory which we have, why do we see more outbreaks of IPH and HHS in the field, is simply the fact that since 15, 20 years we are dealing much more with highly pathogenic avian influenza, more and more with salmonella in certain areas, mycoplasma, which means we keep the breeders cleaner and cleaner and to prevent infection during rearing. So they do not develop immunity anymore until they come into production. And to overcome this, there are the concepts of vaccination, of course. There are live vaccines, killed vaccines, whole virus, and subunit vaccines. This is mainly the experimental trials which people follow at the moment. Live vaccine, there's a live vaccine, 8B live vaccine, serotype 8B listed in Australia. Other than that, there are no live vaccines listed. Live vector vaccines are developed, Newcastle, for example, etc., etc. So what is the concept? If you vaccinate against fowl adenoviruses, first of all, you have to think about your breeder, as I mentioned. This is very crucial. You want to vaccinate the breeder for two different reasons. First of all, you want to prevent vertical transmission if the breeder is infected during production. This is the first argument. And the second argument, what you want to deliver antibodies to the progenies. That's quite important. And then it depends, do you need to vaccinate the progenies as well? Is your immunity enough to cope with the challenges in the field? And the type of vaccine, as I say, in live vaccine, there are different developments. Actually, subunit vaccine is the vaccine we are favoring of. So we are working a lot on subunit vaccines, and on subunit vaccine we work on the fiber protein. You have basically two different compositions of fowl adenoviruses, serotype 1 with a short and a long fiber. All others have two fibers of the same length, but some have two fiber genes and some have one fiber gene. And we started with the one with two fiber genes, which is FADV4 causing HHS, and you can see fiber 2 protects nicely. One bird died. This is the one which had no antibodies. And then we expressed FADV8A, we expressed FADV8B, but to make a long story very short, those fibers do not cross-protect. And that's a great disadvantage. At least in our trial we didn't find that. And that's a disadvantage because we have serotype heterogeneity, as you can see here in red, isolates from Pakistan, FADV4 or FADV2 isolates from Pakistan. And that's why we came up with the concept of chimeric fiber. What we are doing is we cut the fiber at a specific site and we add the component of another serotype. And we call it chimeric or Bragg fiber. And if you do it, for example, for FADV8A and AB, then you can see the antibodies you produce, they are cross-neutralizing. So you neutralize two serotypes with one single protein. And if you use it in the chickens, the solid bars are the challenge controls, whereas the dashed bars are the vaccinated ones. You see protection. And as an example for 4 and 11, histology is a very good example to display protection. You can see this is the challenge control with the lesions in the bursa. These are the vaccinated one, much more reduced the lesions, and this is the control birds. And what we found amazingly is that this kind of subunit vaccines elicit also a local immune response. A local immune response, they trigger a local immune response. If you take the vaccinated only birds in blue and dashed colors, you see in the thymus, for example, CD8 alpha T cells after vaccination, for example, or in the spleens, you can see significant uprise of CD8 alpha T cells. And we think these cells are important for protection. For gizzard erosion, the concept does not work. This is a disease I did not cover yet. Gizzard erosion, production deficiency in broilers. If you see gizzards like this, you first of all think about mycotoxins. that's okay. But you should do histology because you might deal with an adenovirus infection, as you can see here in layers, as we reported for the UK, or in broilers in several countries. And if you do vaccination, you need live vaccination because against gizzard erosion, antibodies do not protect. We infected birds which had antibodies and they were not protected. That's why we use this long time known pseudovirus, which is attenuated. And if you use this virus and you vaccinate, then you can see you have no lesions, hardly any lesions here in one bird vaccinated and challenged. So for fowl adenovirus prevention, it is pretty clear the future will be we have to interfere on the breeder level, we have to vaccinate, but one is the time, when and whom to vaccinate, and how does the monitoring look like, which is very important going along with the vaccination. So I covered 3 different complete pathogens, from a parasite towards bacteria, and the parasite on one stage for attenuated and monoxenic, local culture monoxenic means only one bacterial strain, irradiated metabolically active bacteria and chimeric recombinant fibers. I showed you some efficacy and cross protection studies which were quite promising, I would say. We looked into immunology for all 3 of them, mainly into interferon gamma. And if I say we, then it's mainly those people who allow me to travel. So I'm thankful to the people who contributed, the institutions who support us with funding. And finally, for those who are in poultry research, I want to draw your attention to this journal in poultry research, and I thank you for your attention. Thanks a lot. Oh, just to forget, sorry, I had a final slide and I have 30 minutes left.
30 seconds.
30 seconds, sorry. If you're interested in poultry research, if you're interested in poultry immunology, we are hosting the Avian Immunology Research Group meeting in Vienna in Austria in September, and you're warmly welcome to join with this. Thanks again, and thanks for your attention. Thanks.
Thank you very much, Professor Hess, for your scholarly talk on vaccination strategies for selected poultry pathogens. Now we have a time for a couple of questions. Please raise your hand, and it's better if you introduce yourself first and then ask the question. Can you hand over the mic to him? You have only one mic?
Okay, very nice presentation. We really appreciate you talk a lot about toxin passive, both from poultry industry. No question, just I'll give you 2 comments. You mentioned E. coli vaccination approach. We did a lot of the E. coli vaccination program in the past 30 years, but there can be diverse the serotype. If you use like eradication, the technology, that means you have to consider whether they're able to produce cross protection among different serotypes. That's number 1 comment. Second comment, maybe I would like to remind you, you mentioned the adenovirus. Adenovirus, you use you check the gizzard isolation, or another name erosion. That is, according to our discover our research, we identify bacillus cereus. They were caused the gizzard isolation. This happened frequently in the, particularly in the broiler industry. Right now, we identify there's the prevalent in the yellow fever chicken. So I just remind you, if you check the gizzard isolation, you need to identify whether it is infected by the bacillus cereus. Because right now, there's for the poultry industry, a lot of people are favorable use the probiotics to replace antibiotics. But problem, they have higher contamination of the bacillus cereus. We did this question with Dr. Kashif. We identified this situation in Pakistan. In that back 4 years ago. So I just give you comments.
Thanks. I appreciate the comments, but I want to reply on the comments.
Yeah.
The first reply is for E. coli. I showed the cross protection. We use different challenge strain, not homologous. One is homologous, the other one is heterologous. But it's pretty clear that the field is much wider, and you might have to use, and you should use more of them. I fully agree on that. The second question, I could not go in detail in adenoviral gizzard erosion, otherwise the talk would have been different. The talk was on vaccination. If I would have talked on etiology of adenoviral gizzard erosion, I would have put a slide where you see glycerin, you see fish meal in it, you see Clostridium in it, you see everything in it. What I pointed out is you should do histology. You should do histology, and if it's an adenovirus infection, you should do in situ hybridization. Then you see inclusion bodies, intranuclear inclusion bodies in case of adeno, and you see the relevant lesions coming along with it in lymphocyte infiltration. So I would not say every gizzard erosion, that's not what I said, is due to adenovirus infection, but it's pretty clear, the studies you saw are in SPF birds. It's pretty clear that you can induce it in SPF birds. But I'm happy to give you a talk on adenoviral gizzard erosion and with all the studies and all the details. But thanks for your comments. I think it's important pointing on the diagnostics. Thanks
We
a
have
lot.
time for one brief question. Very brief.
So I have a question about the subunit vaccine. So you have a chimera of 2 fiber proteins. So what do you comment about the cost effectiveness of this vaccine for poultry in future? Would it be possible to meet the cost of attenuated the viral vector vaccine?
I say yes, and you might say I'm naive, but that's the scientific approach. I'm first of all not responsible for the costs, put it that way. Not to think about the costs. I mean, we have now 2 chimera out of 4 proteins, so we have reduced it by 50%. And just to remind you, I also didn't mention we use the baculovirus expression system, but you know there is a company who produces H5 baculovirus expressed. It's on the market. The field is open, I would say.
Okay, thank you.
All right, Professor Hess, thank you very much for your very informative comments.
Thanks.
Thank you very much. We have a little change in the sequence of speakers. Instead of Khalid's name, I would like to invite Dr. Nazir Hussain from CEMB Institute of Animal Health Sciences, Karachi. He will deliver an insight into vaccines and biologics produced at CEMB Institute of Animal Health, Karachi. He has to catch his flight for Karachi, so that's why we call him early. You don't have any flight today. Okay, it's over to Dr. Hussain.
Thank you very much. Pardon me for not to see my old and best friend Dr. Khalid. Instead, I'm here. And number 2, pardon me that I will not be talking much about Dr. Haas has talked about much of research. I have been asked to share the industry perspective, what we are doing in Karachi and where Pakistan is and where we can go about. So might be we can spare some time, maybe next time we can have a talk about research on the vaccine. Today we will be talking about what we do at CEMB Institute of Animal Health Karachi and how we go there. Please. afternoon. And this is, I'm going to represent CEMB Institute of Animal Health, Karachi. And I'm working as Executive Director and Director General for this institute. And actually, this institute was established in 1970 as a part of a Poultry Research Institute, Karachi. And initial funding we got from Government of Sindh and World Food Program, then USAID and the UNDP. And then it was the time when the poultry farming and commercial poultry farming, especially broiler farming, came into Pakistan and it was introduced. So therefore the disease was there. And then finally, the Government of Sindh decided to upgrade this institute into a Sindh Institute of Animal Health, Karachi, whereby we are now attached the institute with the department with the full autonomy and can sell its products in Pakistan commercially, and we can enter into agreement with national and international partners. We are ISO certified since long, since 2006, and we are now CEMB qualified. And then this institute was established by legislation in 2018, and the development is going on, and I think we are now at advanced level of its establishment. Now with the different tasks, this institute was given, that was with the concept of a One Health, what the world is talking about. So therefore, keeping in view the government thought that it's a better opportunity to establish a One Health institute rather than establishing different institute in different area. So therefore, we have now livestock health division, we do have a poultry health division, we have wildlife and pets health division. This is the first time that the livestock department started working on the health of the pets and wildlife. And then we have now fish and aquaculture health division, and definitely the last and the least, that is the veterinary public health division. So these are the 5 health divisions. They are, like a One Health Institute. We have got affiliations with the different universities because having a diversified field in case of a health. So therefore we are having the oldest one, our partner University of Karachi, Sindhi Institute Tandojam, Federal University Karachi, Dadabhai Institute of Higher Education, Federal University, Desto, Salim Habib University, Dow, and Dolwet, Turkey. Now we are in negotiation with the NIG Faisalabad, and then HAG, International Center for Chemical and Biological Sciences, Karachi, and then the Institute of Virology and Immunology, Bern, Switzerland. So, if you see what we are doing this in all health aspects, we are doing vaccines and biological research and production. We are doing essential drug now for the prebiotics, probiotics, and the multivitamins. Serum products like anti-rabies and anti-snake, and then the vaccine drug testing and sales and marketing, disease diagnostic and surveillance, and especially focusing on zoonotic disease, and then meat, milk, and egg, animal origin or animal source food testing. And then we have been given now legislature that we can award now MPhil and PhD degree instead of having. So this is, we are going to open our corridors for awarding degree, and we are getting admissions for the students maybe by March next year. Currently, our structure is Sindh Institute of Animal Health, Karachi, is based on the center in Karachi, head office, and then it's aimed to have different regional headquarters. So it is at Hyderabad, Mirpurkhas, Benazirabad, and Larkana and Sakhar. These are the 5 major divisions in Sindh province where we do have our presence. Initially, we have a surveillance presence everywhere, but the production that is for the public health, especially anti-rabies and anti-snake, that is now at Sakhar. So we have a robust diagnostic and research activity since long, since more than 2 decades we are doing this. So therefore we are supported by different surveillance centers and institutes throughout Sindh province. So how we develop vaccines initially, what we do, that's we have a field surveillance presence throughout Sindh province where we get samples almost on every week from each division. We process them, then we also get some research samples as well. We process them, and then finally we come at a conclusion to the characterization of those and see whether there's a 30% increase in the new emerging infectious disease, or if it is more than 20% increase in the non-matching of the field and the vaccine isolate. So then we bring this to the development section. There's a vaccine development section where we develop master seed development, and then we develop the process how to manufacture this vaccine. Then there's the optimization, and then the test batch and lab trials are there, and then definitely the field and clinical trials are done there, and then it is going to be handed over to the mass scale production and marketing to the CR production line. So what we have currently over the years, that's a poultry vaccine and biologics. We have 64 types and combination. Almost all of the problems what the poultry is facing in Pakistan, we are manufacturing solution for them except Marek's disease. And then for the livestock vaccines and biologics, we have 7 different types and combination currently we are doing. And for the public health products, we have research too, and we are now about to market for maybe within 1 year for the anti-snake one. So to have a bit look upon that, myself I have done the research in the recombinant vaccine production against avian influenza. It was intended that the avian influenza pandemic is coming. So while I was in Germany, I could develop different 19 types of vaccines there for the candidate, for the universal candidate for the to face the pandemic. So it was one of the DIVA and the safest and the single cycle recombinant vector vaccines. So we have different options. And then I came in Pakistan and joined the livestock vaccine department as the director for the vaccine. And then we had now generated here the HSIL, a hydroquelia, that's the new, that was the entry into the hydropericardium syndrome, what the Michael Haas was talking about. And then we had a anti-clone 7, that's live for peacock. And then VVND for Punjab, when we could see in 2014-15 huge infections coming in Punjab. And then we have a different now. The latest one is added into the lumpy skin disease, what we manufactured last year. And then PPR vaccine, which we were more reliant upon the import, now we are manufacturing it here in Karachi, and CCT goats in goats. The next which in the line is anti-rabies, which we are importing from Korea and different nations. And then for the serum production, research has been done on the anti-snake venom sera, and the anti-rabies sera is going on research. Now it's a commercialization for the other mass scale production of anti-snake venom sera in Pakistan. So some of the major products what we produce, that is all the strains of the Newcastle disease. Moctezor, we are more focusing on the backyard poultry and the fancy birds, Lasuta and the commercial broiler and the layer. And the Camorrum infection is usually not there, but sometimes it reappears. It reappeared in Sindh province in some farms of Karachi in 2014. So as a backup, we keep it, and then once it is needed and when we identify the infection is coming, therefore we start doing this practice. We have all the forms, that is in the killed form as well, in the oil and simple. Avian influenza, I think we were first in Asia to introduce this, and this thanks to the PRC at that time. They gave us a task in 1997 to produce H5 and H7 vaccines, and then later on H9. So I think this is the reason that Pakistan is controlling avian influenza largely and not entering into public health. Because the issue is this, that when you are continuously, like the commercial dairy and the commercial layer and the broiler farmers are regularly, like obligatory, they are vaccinating against avian influenza. So therefore the viral load is reducing every day, so therefore there is a little chance of mutations in this way. What we think that this is the blessing for Pakistan that we are doing this, and by having vaccine against avian influenza is actually protecting public health. Then we have a Camorrum disease not only in the live, hottest and intermediate, but also in the killed form in different combination, infectious bronchitis, all in all forms and all combinations what is needed in the market. Then we have a fowl pox. We are the oldest one in the fowl pox since 1970. We are producing it and then we are selling it. Definitely hydropericardium syndrome was first reported in Karachi in 1987. We were first to produce in 1987, and later on we developed Angara disease IL vaccine, which helped a lot in some extent for the broiler production. In 2013 we produced hydroquelia. Now what we see, what we observe in the field, because of the repeated vaccination and evolution of the virus, now the incidence has been reduced from 70% in the broiler industry to 2% in Sindh province, especially in Karachi. So this is one of the success of the story of the vaccines, how you do. And when you do it in a mass vaccination scale, then definitely you are going to eradicate some of the disease up to that level. E. coli, we did it previously for the poultry, because in Sindh province we have a huge humidity, so broiler was facing E. coli infections. We produce vaccines commercially at large scale. Now E. coli infection is not reported as such, so therefore vaccine available there as well. I'm bringing septicemia not only in the aqua base, everywhere in Pakistan is produced, but we also produce an oil base, and oil, there's a septic and montanite oil as well. And then the latest one that was in the series of a livestock that was the mastitis vaccine. So mastitis vaccine, we developed this with the help of University of Faisalabad, and we have a contract research with them. And then definitely we are now having more than 85% of the clinical trials protection and as well as in the field. So mastitis is based on the 5 vaccine candidate vector for Sindh province and 4 in case of a Punjab. So E. coli is not available in Punjab in case of a mastitis, but we have gone through this research. Then Sindh also faced a huge issue of peacock deaths. It was a huge and wide in the media since 2012, and it went on till 2014, 16 and 17. We conducted research, and finally we concluded with the new isolate coming into the peacock, and we have a joint research then later on, and we developed vaccine for the wild peacock as well. And then in 2018 we eradicate this disease, and now peacocks are happy there. And then this next task came, that was in 2022 when the lumpy badly hit Pakistan. Sindh was the first province to import it within no time, and we could control the disease within 3.5 months. And then later on, the task was given by the government to produce its own, because you might have repeated outbreaks in coming years. So therefore, we developed vaccines initially from free isolate, and then definitely got the virus. And then currently our presence for the marketing, what we produce, that is 73% is consumed in Sindh province, 15% in Punjab, 10% in KPK, because some of them is ended at the Afghanistan market, 1% in Kashmir and 1% in Balochistan. And then regarding the public health, we have a 2 tasks that is now recently given, that's regarding anti-rabies and anti-snake venom production. So what we know in Pakistan, that we have 55 different types of snakes. About 13 are venomous and 7 are medically important. Out of 7, they are classified into 2. One is a hemolytic, they are consisting of vipers, and some are neurotoxic, they are consisting of a cobra and krat. They are black in color, you can see here, this cobra and krat. And these are the Rusux viper and soy scale viper and the common vipers. And you can see Pakistan is having a different habitat from Himalayas, glaciers, and coming to the irrigated land, the forest, the desert. So therefore, we have a variety of a snake there. What we do, we have the system of a production there, and this is the laboratory, the latest one. And then you can see that we have a rabies trial area, how we milk the snake, lyophilize venom, and then inoculate into the horses, and we also inoculated in camels. The camels had revealed a wonderful results in case of a serum, in case of anti-snake. So therefore, now we are thinking of in future using more camel, because the size of the protein for the camel, that's antibody size, is a very small, and it have a penetrating quality and issue with that whatever the venom is available with the tissue, that can be utilized in it. So these are different steps of the snake production, and you can see that even a single vial which was produced at our premises during the clinical trials could somehow made this child, I think, recover from that phenomena. And then these clinical trials were registered with the WHO, and they are registered with the clinical platform. We are now developing and working on rabies as well. And we have also, that the government of Sindh has dedicated this island for production of FMD vaccine in future. We are looking for public-private partnership on that area. We have entered into agreement for the vaccine, that is the plant origin vaccine manufacturing, that is developing any vaccines from plants with the Defense Science and Technology Organization, Karachi. And we are, very hopeful that one day we will get breakthrough in this. And then we are developing drug testing and as well as the vaccine testing lab. So this is my last slide, that we are very open, though we are public sector institute, but open to the private partnerships, not only for the contract research, diagnosis, again product development. Currently we are now with the 3 partners entered into this contract research and development. We have a contract or third-party manufacturing for vaccines and biologics. Currently, we have 2 partners in this field. And vaccine marketing, we can outsource vaccine marketing in different products. And then we are looking for partnership in anti-snake and anti-rabies marketing in the future as well. So thank you very much for the questions. Please.
Thank you, Dr. Kabrola, for giving us an overview of the vaccine animal sciences. You also described the R&D activities and production activities. Very informative. Dr. Munir has.
Yes, sir.
Can somebody give the mic?
Okay, maybe I can speak a
All
little
right.
now.
Can you hear him?
Yes, yes, sir.
Yeah, so this is a state institute rather than a private entity. So how you think is everything given free to the public or you charge something?
The other 2 aspects. One thing is this, that in Pakistan, 91% of the farming in the livestock is a very poor and extremely poor people for the livelihood. So the government has taken a task as a public task elsewhere in all the provinces in Pakistan, that if they lose this, then the livelihood will be the problem for the people. So therefore the government thinks it's their task to vaccinate their animals. Those of the 91 segment which is sustainable. So therefore, what we produce, our all development and the salaries, they come from the government, but all the operational costs come from our own revenue. So this is a model that we have about 250 million rupees coming from our own sources as a revenue, and somehow 260 or 70 million coming from government of sources. So therefore, we are like a semi-government. So this public sector is at the behind. We don't make profits. It's only the 10% what we have the cost invested in the cost of production. 10% is only given in the chance for the wastage, or somehow sometime the wage is going to be wasted or you recall the wage. So therefore, otherwise we are non-for-profit. But when they're the contract manufacturing, we fix them and then they are going to aid into the marketing cost and then they save into the market with the different names. So what we have a third party manufacturing, we have a 30% on the label that is manufactured by blah 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 by the institute, and 70% of the label belongs to them.
Thanks.
Yes.
One scientific question, one more.
Yes, sir.
You're saying that you take the serum or plasma from the camel and then directly inject the human for the anti-snake?
No, there were the 2 reasons. One thing is that when we collect the serum, definitely there is all the processes of the sedimentation and then the latest technology of a protein isolation and separation of immunoglobulins and then their purifications, and definitely then there are 2 ways to do this. One is the lyophilization. We also
my question is not that. My question is there not cross reaction because this is an animal antibody and you are giving to a human, it's not humanized.
Actually, what happens that in case of a camel, we expect that there might be some of the people later on when they consume camel meat, there might be certain variations. So it's only the thing which is of concern. Otherwise, in Pakistan or elsewhere in the surrounding, nobody is going to eat the horses. So therefore, the host antigen is always there. When you are going to have a serum, you cannot purify 100% anything. So therefore, there is some sort of allergic reactions coming in future. So therefore, we don't use the sheep and goat and some other animals which are halal for food. So it's only the horse, and now we have started thinking. The blessings and the advantages of the camel serum are overweighing all those dangers. So therefore, we have to tell the people, if you use this camel, then you do not. And we are going to be providing this to that area where camel is not consumed. Usually in Brahmins, the people, they don't love to have camel meat.
No new technology is coming which you can actually.
Yes, recombinant and some protein expressions.
Thank you very much, Dr.
Dr. Kashif Salimi has a question. Please.
I'm Dr. Kashif from University of Agriculture Festival. Thank you for this wonderful presentation. And I hope your vaccine will be registered by the track.
Please.
I'm right. These are okay.
Sir.
Okay, so my part of question is that you do routinely vaccine matching studies. You
Definitely.
update your seed virus.
For all the endemic ones, we do. And for the new one like FMD, we are going to enter into agreement with with the NIPG Faisalabad and the HEJ Institute for International Center for Biological and Chemical Research in Karachi, and we have also Day1 on our right hand. So these three institutes do all the matching studies. But for the endemic one, for all the poultry and for all those which we are doing for last more than 50 years, we do matching at our own facility. So therefore, I started from this. Anything which was in the matching less than 20, more than 20%, we change the vaccine.
Dr. Hess, please.
Yes, please.
Thanks a lot.
Please.
Thanks a lot for your presentation. I was just interested in your comment on avian influenza. You're saying the major point or one of the points that you do not see avian influenza is the widely used vaccination. This for me speaks a bit different to the data which is available from Egypt, for example.
Yeah.
So what's the explanation on that? Do you do obligatory monitoring, and what is the how do you see
Actually,
the
what we do, initially I didn't believe this idea. Keeping in view the Egypt data, when I was working in Europe, working in Hanover, and the European Union was calling that there should be no vaccination. But while I came to Pakistan and worked so far long, that now you see in Pakistan it's almost mass vaccination against avian influenza, whoever it is. So therefore we don't see, otherwise we share all. Egypt might have some demographic and cultural or some other issues, I don't know how they go about and the Egyptian people can tell. But whatever in Pakistan we see the strong evidence that's only available, that is only the mass vaccination. Because you see, we people also love to have debate live bird markets. And I have conducted so many research on this. John Asford, I talked to him and he said Pakistan, India, and might be the China and some other people nurturing new viruses. So I went on this as well. So what I strongly started believing in this, and this is only the factor which I could see that is going to be that when you have a large scale vaccine, like in other cases in case of render-based, like we are doing in human polio, for other cases, it's somehow going to affect the virus replication and reduction. So when you bring the herd immunity, the concept of the same like herd immunity, when you get 70 to 80% of the population immune against certain serotype or against certain viruses or pathogens, definitely the speed or the replication is going to alter. So therefore the availability of the virus for the next host is reduced. So this is how when you have a 90 to 99% of the chicken is going to be vaccinated in Pakistan. So you would see that there's a little chance for the virus to replicate further and mutate further to bring. So because immunity is there available. Please. Yes.
Thank you, Dr. Kalor. I think
Thank you very much, sir.
We don't have any more time for any further questions to this speaker. We have to move forward. Thank you very much, Dr. Kalor. Thank you very much. Have a safe flight. Thank you. Let's move to the next speaker. Next speaker is a wonderful scientist, he has been working at NARC and he got early retirement in 2030, and then he started his own Grand Pharma. He has a state-of-the-art R&D and production facility in Punjab. The day before yesterday, he organized a seminar by Professor Munir Iqbal. I was also in the audience, and I hope Dr. Munir Iqbal, you are going to deliver your talk tomorrow. Yes. Will it be the same talk you delivered in the seminar? Okay. All right. So it is over to Dr. Khalid Warta. The title of the talk is Suggestion in Indigenous Vaccine Development in Poultry Setting.
Thank you very much. And first of all, I'm thankful to Dr. Kaloro. He has covered some few things which I wanted to say, so it'll save me some time. Basically, I'm also not going to present any specific scientific data, because rather I would like to have an overview of how the vaccines in poultry got developed over here in Pakistan. And maybe answer at some stage what Professor Hess has asked. So maybe we'll be discussing that part, why influenza is not reported from Pakistan. I think as we all know as professionals, there are two major challenges. One is the development of antimicrobial drugs, the resistance injection, and development of more effective vaccines. That's the two target. Because today we are mainly focused on the vaccine part, but I think all of us understand that because of the availability of the generation of resistance in microbes, the vaccine could be a better option to go. So that's one of the reasons writing it together. Sorry. Just to have a look, what's the population of animal, livestock and poultry currently prevails in Pakistan. I'm because trying to focus more on poultry, but just for the sake of reference, I've just pointed out here that these the population's size that it basically indicates the requirement. The focusing on poultry, this covers basically the grandparents, the broiler breeders, layers, and broilers all together. And if we talk about the coverage, basically it's very difficult to find out coverage of each and every vaccine. Generally speaking, if I say generally, it's a big range. When we talk about livestock, Professor Dr. Kalodo has actually gone, maybe some other person from livestock is here. We are talking about very few vaccines which we can claim coverage more than 50. Most of the vaccines are really coverage very less. So this speaks not about the quality of vaccine, and it speaks about the quantum, the production capacity as well. So similarly in commercial poultry, because it's all, I think this one is free, the poultry is all payment basis. So I think that's another part, although it's expensive, but everyone makes sure that all type of vaccines which are required are carried out. Rural poultry, it is mainly focusing on a couple of diseases, and then again, it's the government is giving free vaccine for this backyard poultry, so that takes care of some of the components. But very few people in the private sector, not the private, the backyard poultry, will be using the vaccine by paying anything. Similarly, the major diseases, I remember one of our colleagues would say many years ago that there's no disease mentioned in the book which is not present in Pakistan. So we have reported number of diseases at different times. So I think I've just listed down especially poultry diseases, grouped them in the viral diseases, and this is the bacterial disease which we have in this country. I'll stop and commenting on the other part at the moment. Now about vaccine market, if you see the local production, local producer, they're very small share. I think we have only 2 companies in Pakistan, including Grand Pharma, where I work, which are really producing vaccines locally. These government institutions, they do have component, but most of their share goes to the either backyard poultry or the broiler farmers primarily. Breeders and grandparents and other sensitive, groups, they do not use these vaccine from government institutions because of the SPF Acts and other issues prevailing. Anyway, the imported share is major, and this includes not only those vaccines which are produced locally, which are very few, but it covers all the vaccines that they use. It means the Newcastle, even if it's produced in the country, still a huge quantity is imported. So that's another discussion, how can that be overcome? And similarly, livestock, as I said, it's a current share is very well based on different vaccines. We have more requirement for LSD and foot and mouth as compared to other vaccines. But the coverage is more in HS vaccine, for example, which is locally produced. But other many diseases which we do not use vaccine, like VVD, IVR, and very few the commercial farmers, big farmers, they selectively use it. And most of those vaccines are not even registered in the country because the market is not present. So this component looks small, but the requirement is very small as far as those livestock is concerned. This is again not used over here. Nobody is producing it, and anything required in pets and other market, they are all imported. Because my basic the concept, the purpose of discussion is just to ask the question, really, why do we need to have local vaccine production? So this is kind of few arguments which we always discuss, and I think most of people sitting here would understand clearly that it's the availability is one concept. The other one is cost effectiveness. And the effective disease control plans, which when we had the, like example, history of rinderpest, so there was the controlled disease program, and now the PPR is going on with the help of FAO. That's again disease control program. So these kind of programs can only be run if vaccines are available in sufficient quantity and continuous flow. Otherwise, even running those control programs, we have to rely on all the imported vaccines. So that's kind of one of the reasons to do it. We have done it successfully in case of poultry for avian influenza, so I'll be discussing the next slides. And the whole theme was there. We prepared the hydropericardium vaccine in this country, and there's no vaccine available anywhere in the world. But similarly, we have the influenza H7 vaccine produced over here. Nobody is producing it abroad. So that's, I think, one of the maybe good things happened that we could save it. So to acquire the desired efficacy, that's another point to discuss. The homology of the organisms. So if we are doing it, characterizing it ourselves, we can have a better match of those vaccines. So that's one of the major concerns nowadays. When we are using vaccine for the past 76 years, a huge number of vaccines are being used, maybe imported or local, but mostly, as I said, imported. But diseases, I think no disease has been controlled yet. So that's one of the arguments is that our vaccines may not be compatible, but we really need. And third, the last point is we can develop vaccine on an urgent basis in case there's a mutation or there's a change. So that's why I think we need a system within the country whereby we can devise, develop and propagate our local vaccines. A few things about the historical perspective. I think as mentioned by Dr. Carollo, early I just divided it into two, like pre-80s era and post-80s era. In the early days, when the poultry sector, the commercial poultry sector started in the 70s, 60s, mid-60s, we were only using Newcastle and fowlpox vaccine. And the institutes are built, this institute that Dr. Carollo was mentioning, Karachi Institute, that was first started vaccine, producing local vaccine. And those vaccines were based on Newcastle, fowlpox, and IBD. And later on, the institutes were expanded, as you heard in, earlier. Now they're producing a variety of other vaccines as well. This era is basically goes to 88 Angara disease, situation. I know Dr. Hess is quite senior like myself. We have worked together on Adeno4, so we know the history behind it. But it's maybe interesting for the young guys sitting here that in those days, the poultry was at very early stage. We didn't have good diagnostic labs. We didn't have a good vaccine manufacturing units. But then this disease comes, this broilers, and the adenovirus kills 70-80% mortality, and nobody knows what is it because of the shortcomings. Eventually, the scientists together, groups from NARC, very active, PRI Karachi, PRI Lahore, all these people get together and had a guess, So imagine that we had some AGPT test carried out and just determined that it is adeno. That's it. Couldn't type it. So urgently the old days, we blended the liver, produced some and vaccine kind of autogenous vaccine, and luckily to our surprise that worked. At least we could save our industry at those days. I'm not talking here about the argument of good vaccine and bad vaccine, but in those days I think that was basically the initiation of this concept of developing vaccines in case there's a disease which is not prevalent reported anywhere else in the world, and you come up with that situation, what would you do? So that's one major reason I think about having some setup where you can really encounter that challenge you can handle. Next thing happened in 1995 when we first time avian influenza. Similarly, at least I can say people at my age at that time, none of us had seen influenza before. I've never studied it. And I remember when I was studying PhD, one of my technicians, when I was coming back from USA, she wanted to give me some antigen to carry it home in case we need to research, because countries like us take a lot of time to bring those materials. And she forced me to carry some polyvalent antigen and anti-sera for influenza. And I was laughing at her. we never had it. Why should I carry it? But can you believe those materials which I brought helped us to diagnose first 1995 outbreak, just per chance. So that was the situation. But again, I always say, maybe blessing, no vaccine existed in those days. So again, we had experience of adeno. We did the same thing. We could grow it in eggs. So we are in Karachi and we are in Lahore. They were in the upfront. We were producing, the seeds at NARC, doing challenge studies, and eventually together we were able to basically develop this vaccine. And very interestingly, it took us just 6 to 8 months, almost a year, we could control this influenza spreading from one of the places from where it started. So very northern part in Islamabad. So that's not a historical perspective. Next thing came in 1999. We have another strain, H9, and this time we had experience. So we actually basically, although moved fast, but now this was a different kind of virus. And then again, I said in those days, lack of knowledge, we didn't know much about the low path and high path and mutational strain and all those things we now know. So it took us a lot of time to try to control it, but it could not be controlled properly because of the low path. And as I said earlier, that the previous outbreaks were controlled with a strict policy of vaccination and culling and compensation. These were the three things we did. Early reporting, early detection, and then if you do that, then you get compensation for your flock. But you have to have vaccination. Once you have declared that vaccine is been prepared, you have to use it. So the next outbreaks were handled with this formula. So we were able to control this at early stage. In this case, because of low path, so many people didn't, you can say, give it much thought. So the disease prevailed, and as the virus gets settled, it keep on starting different groups of mutation at different places, and that's what's happening since then. Genetic mutations are occurring, new variants evolving, vaccines are failing, so that's kind of part of the whole story. The next thing happened in 2003, another outbreak at CEMB occurred after 1995. Again, we had the local vaccine prepared again, new strain, new vaccine was prepared, and we were able to basically control it. And the last thing happened in this story was the H5N1 2006. Again, many people ask us questions when we report it, when we have published many papers, data is available. On ground reality is that we were able to control it in 2008. And if you go back to that time and just take the data and examine all the things, we followed a set pattern as a policy. We are going to vaccinate. And in those days, many European and American authorities, they were against vaccination. It's okay, every one country has got their own limitations. But now just in 2022, in European, the WOW has said yes, you should be vaccinating against influenza. So it's okay. But I think we did this practice basically earlier, not today. We have been doing it for the past so many years. So since then, our policy is to vaccinate the high-risk birds, like the grandparents, parent stocks, and parent layer with the vaccine. There are 3 to 4 shots. They are monitored quite regularly through post-vaccination monitoring. Antigenic, compatibilities studied. And any isolate once in a while, we have some wild H5N8 outbreaks, very limited form 1, 2. We applied the same policy, culled the flock, compensated it, destroyed everything, and it didn't spread. So we should have to take this, I think, pride that we were somehow able to continue this strategy, and still today, with H5N1 going around at other places, Pakistan is still safe till to date. So, outcome of this whole effort is that we had a successful control of adenovirus, the Angara disease, and also Pakistan got free from all these 3 menaces of the influenza. Now, just brief about the local vaccine scenario. I think this is the place Dr. Kalora has mentioned, the institute. Yes, there are some government institutions. They are producing some poultry vaccines, and then there are private companies. 3 of them are producing a number of vaccines and poultry vaccines locally. And the new technologies have been used at certain places of the vaccine matching, as Dr. Kalora has mentioned. We also do it in our own capacity. There are the national, at NARC, there's National Reference Lab for Poultry Diseases. They are involved in doing surveillance and monitoring and sharing the data about any mutational or sequential data. So that helps us to devise the vaccination strategy. The technologies of new technologies are being used to develop master seeds, and those are being used in collaboration with certain institutions, and that helps us to improve our vaccines. The technique of cartography is adopted, as I mentioned earlier, to select the seed of your choice, the virus of your choice for vaccination. Subunit vaccine are also being carried out, especially for adenovirus. We are producing, one of the group is collaborating with us also, we are producing that vaccine. And similarly, the multivalent killed vaccine is used through the reverse genetics technique, the seeds are developed, and this is one of the pictures just to show him selection of the seed. This is the list of the vaccines produced locally. Dr. Kalora has also mentioned it. A number of products are from his institute, but this is also produced in private sector. In private sector, this one institute is producing a couple of live vaccine. Rather, most of the institutes are producing killed vaccine. And these are the list of other diseases against no vaccine is being produced. So these are our basically, I'm not saying that because this is being produced, then we don't want to import it. This is being produced for very small quantity, and they cannot fulfill the demand. So the point is that we can make it. If we can make it this one, we can also make it. So it needs few things which I will be submitting in the next couple of slides. I think there's a major issues right now for the vaccine production, deficiencies, which is we have some outdated vaccine production facilities at most of the places. Especially the public sector places, the institutions are pretty old. They need to be upgraded, and the vaccine system needs to be more functional from at national level. Individual laboratories or individual pharma unit may not be able to perform this task. The cost of the things we are importing is increasing day by day. So we need a policy from the government whereby they can have some kind of support for the establishment of these production units by utilizing these biotechnology related equipments, biofermenters, and that kind of technology. It will come only with certain regulatory mechanism devised by the government institutions to support the local industry to expand it. Similarly, human resources need to be strengthened. This vaccine is very kind of like an isolated activity being carried out by very few institutions. So there are very few experienced experts available in this area. Same is a deficiency in the public-private collaborations that's required, and I expect from this conference when we end the recommendations, these kind of points are also considered, which are handicap for doing the private sector production of these vaccines. There's low R&D conversion to product. If you see the universities, their projects, their PhD theses and all those things, so there are huge data is available. It needs to be converted in products, and that can be done by collaboration through private sector. So we have to devise a policy. And the last thing which may look painful, unfortunately, but the minister is not here, otherwise I could have explained to him as well, that vaccine production is not priority by any means of the government, may it be human vaccine or animal vaccine. Sorry to say that, but that's reality. And despite the fact that we are talking about AMR, and despite the fact that FAO, WHO, everybody says replace these antibiotics with vaccine, we can do it in a good job in case of animal health. I'm very much convinced that we can do it, but we can do it through vaccines. And that will be long-term solution. I hope that attract the policymakers. This is just last two slides. Way forward, again, we have, as I said, less knowledge about the antigenic variability of local pathogens. So I'll encourage the R&D organizations. They should come forward, identifying the different vaccine candidates, and that can be done through public-private collaboration. Vaccine research and development may be made a priority area, as I said earlier, and there need to be, from a private perspective, I'm saying there need to be special tax exemptions which can help to bring in technology from abroad in terms of equipment, in terms of adjuvants, in terms of other technology which is available, and it is for sale. It's not that's not available and cannot be brought in. It's because of our policies and our I would say unfriendly policies which hinders us to do these kind of things. And that's kind of we need to improve our import part by putting some similar restrictions which you put for local produce goods. So they have to go through similar channels. If you encourage import, you should also apply those requirements which you expect from the local producer. So they need some more restricted, technically a restriction on imports, which are technically irrelevant vaccines, or those strains should be somehow inhibited to enter to the country for not only helping the indigenous vaccine producers, but also holding back the new intent of new organisms. Similarly, we think that the imported vaccine needs a vaccine matching studies as well before allowing it to be registered in the country. The existing vaccine testing capabilities at our regulatory laboratories, NARC, at National Veterinary Laboratory, need to be enhanced because these laboratories were doing the job in the past 2 years and their funding issues. But I would love that if they could help us to test these imported vaccines and local vaccine, whatever, and set a standard for that. And by doing all these policies and by considering all those aspects, maybe we are able to do some JVs with good international organizations. There are some friends coming from China sitting here. Yeah, we have already had meetings, number of people coming interested to do investment over here. But unfortunately, due to the lack of our coordination, I think the very stringent policies, or rather no policy of encouraging the JVs, we are still at very early stage of acting. Thank you very much.
Thank you, Dr. Khwaja, for a wonderful talk.
Thank you.
And you have rightly mentioned that vaccine production is not the priority of the government. There are several other things which are not on the priority list we will discuss later. But you are doing a wonderful job, and in the morning session, Minister Saab also mentioned that there are many private sector investors and entrepreneurs who are willing to produce so many vaccines. You have the capacity and the infrastructure. You only need the buyback guarantee from the government. Even the government is not interested to go into that venture. So we have noted your questions. I will convey it to Minister Saab and some other authorities as well. Now the talk is open for discussion. We have time for a couple of questions. Professor Salimi.
Sir, as you have already covered in the last part, but I need your comment on the joint ventures, because I think in Pakistan, like in Fedme, we have a government law that foreigner can invest, but he will be the 49% share, and Pakistani should have a 51% share. So therefore, that factory or production unit will be registered in the name of Pakistani citizen. So do you think it is possible in the poultry vaccine sector, because you have a very strong collaboration with Chinese partners and they are also interested, maybe they can go for the export of vaccine from here with the joint venture and we can get vaccine at comparatively less prices. Is it possible?
Yes, it is possible. There are so many other things along with it. It's just one point you are mentioning. Just having share and having responsibility is one end. Second part is basically the restrictions or handicaps with the trap registration issues in both, for example, our airports, we don't have a cold chain mechanism at the airport. So, I mean, it's very difficult to rely on somebody producing vaccine in Karachi and bringing it to KPK or somewhere. So these kind of issues are there. So when you are in, obviously, other these helps, or you can say regulations or arrangement will have to be made. It's very easy to do some production and export it in some cases like cotton or something. In vaccine, you understand there are a lot of other standards. Similarly, the restriction on the import of those eggs, SPF eggs, they are considered as hatching eggs. So they put a price, the very expensive tax rate, they think we're going to get a chick out of it and chick is going to be sold at this price. You keep on telling them it's just a fluid and it may not even pop up, To rules are rules. So these kind of small things, I want to get into details. There has to have some very clear policy. Otherwise, just saying that you allow a joint venture and that won't help.
All right, Dr. Khwaja, thank you very much. If there is no more questions, we can move to the next speaker. The next speaker is Dr. Jao San. He is the Additional Director DRAP and Chairman Gap Analysis Committee on BRI's Chairman CALSD. He will talk about Drug Regulatory Authority, Pakistan, and its current operational goals. Dr. Hussain, you have 15 minutes, please.
Assalamu alaikum. My name is Dr. Jas and I am working as the Additional Director of DRAP and Chairman Gap Analysis Committee on BRIs and CALSD, Committee on Availability of Life-Saving Drugs. So DRAP has its organizational structure. Its head office is located at Islamabad, and there are different regional offices at Karachi, Lahore, Peshawar and Quetta. We have one laboratory at Karachi, which is called Central Drug Laboratory Karachi, and the second one is at Islamabad, which is called National Control Laboratories for Biologicals, to test the vaccines and all other biological products. Our vision is health for all 2025 through access to medicine, contributing to advancement in universal healthcare system in Pakistan. And our mission is to ensure the access of safe, quality and efficacious medicine and therapeutic goods at affordable prices to the common man of Pakistan. We are working as a highly professional and world-class regulatory organization at par with the international standards and best practices through effective management, regulation and enforcement, and to support national health system management through effective therapeutic goods management and strategies. So currently, we are going towards automation, digitalization to enhance the transparency. Then there is evidence-based decision-making, regulatory reliance. Then we are collaborating for the purpose of harmonization to the international agencies like World Health Organization or PICS certification. And there is an ongoing continuous process which is going on in the DRAP. This is the organization structure of DRAP. We are working and currently under Ministry of National Health Services Regulation and Coordination. And this part, we have the primary, different health departments at the provincial levels which regulate the sale of the drugs, whereas at the central level, Drug Regulatory Authority of Pakistan, it deals with the manufacturing of products, pharmaceutical products, definitely including vaccines, biologicals, imports, exports, prices of drugs, and overall quality assurance as a form of national task force. So these are the federal subjects. So DRAP organogram is, there is a policy board at the which issues various policy guidelines to DRAP time to time. This board is headed by the federal concerned Secretary Health. However, in this board there is provincial representation from all the provinces, and various technical experts are in this board which gives the policy guidelines to DRAP time to time. Then under this board there is Chief Executive Officer of DRAP, and then there are 13 directorates of DRAP, including Director Registration, Director Licensing, Biological Products, Health and OTC, Medical Devices, Controlled Drugs, QA, Pharmacy Services, Legal Affairs, Administration, Costing and Pricing, etc. These are the 13 directorates. So there is this exclusive directorate for the biological products, and the government is always facilitating the licensing and registration of vaccines, anti-seedar and other biological products expeditiously. Currently, I think there is no application as such is pending on the part of DRAP, and if there is application is pending, there may be some shortcomings which are immediately communicated to the quarter concerned. So our mandate is the human drugs, pharmaceuticals, human drugs, biologicals including vaccines, health and beauty products जो कि पहले नहीं थी. These are alternative medicine, homeopathic, ayurvedic, Unani products. Control drugs, there is specific division which deals with the narcotics and control drugs. Then medical devices, veterinary products and veterinary pharmaceuticals. And also there is a separate directorate of pharmacy services which deal with the clinical trials. It deals with the research organization and various protocols are laid in this directorate. This is our legislative framework. That first there is licensing and inspection, then the registration and enlistment, then surveillance and the vigilance. So if any company want to get the registration of vaccines, first of all there is licensing and inspection. Then subsequently registration is conducted, and afterward there is the surveillance of quality assurance of that product which is being manufactured for that particular firm. Then vigilance is being done by our provincial partners through National Task Force. So currently UAS has taken the lessons of FMD vaccine by following this procedure, and subsequently they have also got the registration of FMD vaccine, and now they are manufacturing this vaccine for the local population. RAB operation have, this is, first part is the licensing, second is market authorization, the third is quality assurance, as I told you that there are GMP inspections, then the import-export controls, then pharmacovigilance is being done by the DRAP field offices as well as the provincial governments. As far as vaccines are concerned, there is a lot of the system of biologicals which is being done by the NCLB, National Control Body, which is located at Islamabad. We have the quality control testing, risk-based sampling is done by our field force, then Central Drug Laboratory Karachi or the NCLB, Central Control Laboratory for the biological, they test those samples time to time. And if there is any discrepancy, then the case is proceeded accordingly. Then clinical trials, as I already told you, that there is clinical studies committees which deal the licensing of the central research organizations. The regulatory aspects में there is a current status that our DRAP is ISO 9001 to 2015 certified. Then we have notified biostudy rules 2018. These are all on the website of the DRAP. Then ICH guidelines and common technical document is being followed. CTD on the format of CTD. Then we have the GMPF schedule B2 notified rules, drug licensing, registering and advertising rules 1976. There are 2 labs which are under further strengthening. Market surveillance, already I told you that random sampling, risk-based sampling is being done, then surveys are conducted and various inspections are done which are being monitored by the DRAP consistently. Then there is vigilance by initial stages of the post PV system. The future implementation is we are going towards the WHO QMS implementation to attain the level 3 of WHO certification. Then ICH standards, bioequivalence study working is being done on bioequivalence studies to pre-qualify the various bioequivalence centers so that the products which are being registered, they first go there for the bioequivalence studies and submit their report which will be considered by the registration board. Then PICS and GMP quality risk management approach, WHO pre-qualification of CDL and National Control Laboratory. So our provincial laboratories, there are 5 laboratories in Punjab which are WHO pre-qualified. And now we are working on the WHO pre-qualification of CDL and NCLB, which will be done very soon. Then there is a discrete system with risk-based post-marketing surveillance and the full implementation of the pharmacovigilance. So we are at DAB is at present it is in harmonization with the various international organizations. As I already informed you, like WHO organization, World Health Organization, we are going for the global benchmarking maturity level 2. Similarly, we have 5 WHO pre-qualified laboratories already, and we are DRAB is going for the further WHO pre-qualification of its 2 laboratories. Then PIC certification, this system is also going on so that the DRAB will make the collaboration with those regulatory agencies which have the stringent regulatory bodies. So by the PIC certification, DRAB will come in the that group, and the exports will be increased. This is the market size. So for the WHO assessment, various institutional development plans have been given in 2019. Then in 2020, 23 institutional development plans were identified by WHO, which were done. Then in 2021, 9 teams were notified in the DRAB for this WHO pre-qualification, and a verification report was generated in 2022. And in 2023, there is formal benchmarking. Observe what it was conducted by WHO. So DRAB is going to achieve the WHO benchmarking level 2 and subsequently level 3. So our core values are the automation, transparency, evidence-based decision making, regulatory reliance, as I already told you. So I want to tell, this is our future goals to effective regulation through harmonization and cooperation with the memorandum of understandings, WHO GBT level 3, QC and quality assurance approach, implementation of the quality management and risk management approach, and the PICS membership. Then quality surveillance, early access to new treatment and addressing shortages, and strengthening the pharmacovigilance. These are the various aspects which are being dealt in the DRAB. Safety communications, stakeholder engagements, rational use of medications, and access to information. It is already done in the DRAB. So our new initiative which are being done, has already been done is the automation in the DRAB. We are now the paperless agency, and there is no hard files, there is an electronic filing system, and then there is an integrated computerized system for filing the applications of registration and the licensing. And every firm, rather of the private sector or the public sector, they apply online, and those applications are speedily and effectively with the timelines they are being decided. So these are the digitalization, it is the various initiatives are there in digitalization. As I already told you, Pakistan integrated regulatory information management system is working on online import and export. Application system is going on, then track and trace, DRIS system, medical device, establishment licensing system, then VigiFlow database for adverse drug reactions, and online NOCs and permission for import of personal use. So our whole system of the DRAB is now digitalized. If a person who don't even have the access to medicine in Pakistan, across the country, he or she can apply online, and within 24 hours, he or she can purchase any medicine from any country of the world. So this digitalization has increased the efficiency of this organization, and as discussed earlier, that government is there is some observation that the government is not working on the particular vaccine. Though I told you that being chairman of the gap analysis committee on VRIs, we are consistently engaging with the various VRIs, and they are under the process of licensing 6 VRIs in Peshawar, Karachi, Lahore, and their continuous our collaboration has made their system very fast, and they are getting the licensing and registration on very fast track basis. If there is any shortcoming, we communicate with them, and we are completely day-to-day basis. We are engaging with them to get the registration and licensing of the vaccines at VRIs.
Thank you. I'm afraid we are running short of time. You have to conclude.
Okay. Now these are the various procedures to obtain. These are available on the website to how to obtain the license for the vaccines and biologicals, and various instruments which are required for the quality control, and what are the things which are required to be maintained. So thank you very much for this.
Thank you, Dr. Hussain, for enlightening us on the working of the draft in Pakistan. We can have just one quick comment or question. Just one, very brief.
Thank you very much for this wonderful presentation, Dr. Zia. I just have one question about the clinical trials. I'm Dr. Zaida Fatma. I'm working as a program leader for National Respiratory Proportion Disease. So I'm from the vet side. So I have a query that from where do you do these clinical, because I'm an epidemiologist as well, from where do you do these clinical trials if some veterinary vaccine is coming in the country or some veterinary drug is coming in the country, and in which lab do you do the...
There are various center research, various organizations which are pre-qualified by the draft. We have our one directorate, which is a pharmacy services directorate, and there is a clinical trial committee under that directorate. If someone import anything and want to do research on a particular product, first he or she or applicant has to register that that organization where that research will be conducted. Now there are various, there is a checklist and various documentary requirement which is available on the website. Once the application is received in the our pharmacy services department, then that clinical trial committee or board that deliberate on it. If there is any shortcoming, they will discuss with the applicant, and if that application is complete, then that CRO is approved to conduct that clinical trial, and the protocols are also approved.
Thank you.
So this is the procedure.
Dr. Sam will be around. You can ask if you have any questions. Please hold your question. You can ask during the session. Now we move to, thank you. Now we move to the last speaker of this session, and after this speaker we have some good news for you. The organizers intend to serve your team. Let me introduce Dr. Wasim Abbas. He is Principal Scientific Officer at National Institute of Biotechnology and Genetic Engineering. He has 26 years of experience in pharmaceutical sciences, specializing in molecular biology, virology, and cell biology, holding a PhD from the University of France, with notable contributions to HIV, latency, and cytomegalovirus and organic phosphates. He will be talking about opportunities and challenges in foot and mouth disease, vaccine development, and commercialization in Pakistan. Very interesting topic. It's over to you, Dr.
Thank you, sir, for a nice introduction. FMD is endemic in Pakistan and causing a huge economic crisis in the dairy sector of the Pakistan. According to the studies, approximately 35 to 40% of animals are exposed to the FMD before they reach at the age of 1 year. And the annual losses due to the FMD is approximately $693 million in Pakistan. So in this regard, I will discuss the opportunities and the challenges in the FMD vaccine development and commercialization in Pakistan. So little bit introduction about the FMD. FMD is a highly contagious, infectious, and a devastating disease of cloven hoofed animals. There are 7 serotypes But in Pakistan, there are three serotypes: serotype O, A, and Asia 1 is present. It is very difficult to talk about the FMD in front of Donald King. He's already explained a lot of things, so I will try to cover briefly. So I will discuss about the FMD. FMD is a single stranded positive sense RNA virus. It encodes the structural and non-structural proteins. 4 structural proteins: VP1, VP2, VP3, and VP4. And the 1 unit of VP1, VP2, VP3, and VP4 form a protomer, and the 5 protomer form a pentamer, and 12 pentamer form the capsid. It means a single virus contains the 60 copies of VP1, VP2, VP3, and the VP4. Regarding the non-structural proteins, 10 non-structural proteins. These non-structural proteins are important for one application. Non-structural protein encodes the 2 proteases. One is the liver proteases, and the second is the 3C-like cysteine proteases. Liver protease is important for viral pathogenicity. It shuts off the host translation machinery. It inactivates the host immune defense system. Regarding the UTR regions, FMD 5' UTR region contains the 5 segments. The first is the S fragment, then the poly-C tract, pseudognode, cis replication element, and the internal ribosome intricide that is important for the viral translation. The 3' UTR contains the approximate 90 nucleotides, a stem loop structure, and a poly-A tail. If we talk about the FMD disease, no doubt the mortality is less, but the impact is huge. Here we can see the losses due to the FMD is direct and indirect. In case of direct losses, we have loss of milk yield, loss of weight gain, loss of drought power, and the death of the animal. In addition to the invisible losses, we have the fertility problems and change in herd structure. In case of indirect losses, when we have the disease, we have to vaccinate the animal, that will be cost. Then we have to restrict the movement of the animals. And then the most important is the access to the market. These are just examples. We can see the UK outbreak and the outbreak in Taiwan. There are only 5 companies that actually produce the FMD vaccine, that is the Biogenesis, Vago, Marians, MSD, and the Vedans. And the total market is 1.4 billion, that could be doubled in 2030. Now we discuss about the FMD vaccine in Pakistan. The annual losses due to the FMD is approximate $693 million. A small amount of vaccine has been prepared by the veterinary research institutes against the demand of approximate 300 million doses. And the large amount of vaccine has been imported from China, Russia, and Turkey. So there is a huge gap between the demand and supply of the FMD vaccine in Pakistan. If we talk about the FMD vaccine, NIPT is working in 5 areas of FMD research. First, we perform the isolation and the propagation of FMD vaccine cell culture. The second area is the molecular characterization. And then third area is the adaptation of FMD into the cell culture. And the fourth area is the vaccine matching. Just to compare, we have performed the vaccination of FMD vaccine via Lahore, RI, FMD vaccine from Marion, and the locally produced FMD vaccine. These are just examples and rough data of the vaccine matching. For example, we raised the anti-sera of 4 vaccines: Nifty vaccine, Viral Lahore, Mariel, and the Raya vaccine, and then cross-checked with our local isolates, that is the Pan Asia 2. Here we can see the neutralizing titer of the Nifty vaccine, that is approximately 1 to 64. In case of locally produced vaccine, we have the very low neutralizing titer. In case of Mariel, it is approximately 1 to 8, and in case of Raya, we have the same neutralizing titer. Then we perform the vaccine matching against the serotype Asia 1, and these are some results of the cross match of the locally produced vaccine and the Nifty vaccine Mariel Array against the Asia 1. Here we can see the neutralizing titer of the Nifty vaccine is approximately 2 into 256, where in case of Viral Lahore, we have the very low. In case of Mariel, we have the neutralizing titer approximately 1 to 64. The same in case of Array vaccine. Then we perform the cross matching with the Iran serotype A and the lineage was Iran 05. And we check the neutralizing titer. In case of Nifty vaccine, we have the neutralizing 1 to 1024. In case of locally produced vaccine, we didn't get any neutralizing titer. In case of Mariel, we have the good neutralizing titer, but in case of Arya, we have very low neutralizing titer. So we routinely cross-line the commercially available vaccine and perform the cross-matching studies. In addition to the vaccine matching, the fifth area of research is the vaccine development. And we produce the FMD vaccine at the lab scale. And we recently performed the small clinical trials and estimated the neutralizing titer of Nifty vaccine with the already available commercially available vaccines. Now I will discuss the challenges of FMD, why it is difficult to control the FMD vaccine. Virus is very difficult virus. It is not an easy virus to it replicate very fastly. Large host range and the antigenic the diversity in the antigens is very high, and there is no cross protection among the serotypes. And the second point is the vaccination failure, meaning the vaccine does not match with the local isolates. So it's very difficult to control the vaccine, to control the disease with the vaccination until it is matched with the local isolates. Then there is no strict quality monitoring of the vaccine, and there is no control of movement of animals. If we see the progressive control of FMD pathway, we are at very initial phase of control, meaning we are just on the surveillance. We have not the strict rules and regulation for the vaccinations. The reason behind this is that in Pakistan, the FMD policy is mostly toward the cattle and buffalo. However, sheep and goat are also good source of FMD, means they act as reservoirs. They act as a source to spread the virus to the cattle and buffalo. If we like to eradicate the FMD from the country, we have to vaccinate the sheep and goat as well. Then there's no support from the government. Problems in the clarity and control and responsibility of R&D organization and the organization working the under the umbrella of provincial governments. Biosecurity and containment, there is no strict regulation to control the virus. And while vaccine availability means there is a huge gap between the demand and supply of FMD vaccines. There is no available of human resources and already support from the governments, and there is false statistics about the disease and the vaccination in the country. What is the way forward? If we would like to control the FMD or the eradicated FMD, we need 300 million doses of FMD. For this, we require the good infrastructure. And this is the example of biogenesis that are producing 250 million doses of FMD per annum. We have to establish the FMD antigen production in suspension culture using the bioreactor. And for this purpose, we require the trained manpower. In addition to the trained manpower and the suspension culture, if we would like to produce the FMD vaccine at good price, we have to indigenize the cell culture media and excipient which are used in the FMD vaccine production. Otherwise, it was not affordable for the our community. So it's all about. I would like to acknowledge Nifty and Pakistan Government Institution and the CEMB and the Punjab Agriculture Research Board for providing a project on the FMD.
Thank you, Dr. Hussain, for a very good presentation. Now we can have a couple of questions. Anybody has a question? Okay, if there is no question, even you can ask during the tea session. Thank you very much. By this, we conclude the first technical session. In the session, we have 5 speakers. There was one preliminary talk, scholarly talk by Professor Michael Hess on vaccination strategies for selected cohorts with pathogens. Then we had the keynote talk on progression in indigenous vaccine development by Dr. Khalid Naim Khaja. So where is he? He's just all right. Okay. So the only progressive private producer of vaccine in Pakistan. And the other speaker was from GRAP. Thank you, Dr. Hussain, for enlightening us on the working of the GRAP, certification, registration, etc. The last speaker was Dr. Wasim Abbas. Thank you, Dr. Wasim, for a very good talk on opportunity and challenges in foot and mouth disease vaccine development and commercialization in Pakistan. I hope can conclude all the sessions tomorrow. We'll sit together for a discussion session. Okay. Yeah. We will discuss tomorrow or tomorrow. Tomorrow. All right. So from this session, it's over to you now to announce what announcement you have.
Thank you so much, sir. First, I would like to invite the director CEMB to please come on stage and hand over the souvenirs to the speakers and the chair and co-chair. May I request the chair and co-chair to please join Professor Dr. Moaz on the stage. Dr. Zia Hussain, is he there? Dr. Zia Hussain.
I think he will come tomorrow.
Dr. Wasim Abbas, so please receive your shield.
Very good.
There is souvenir for Professor Dr. Abdul Haq sir. For co-chairing the session, please give a big hand to Professor Dr. Abdul Haq. Professor Dr. Shahid, they will again receive the shield. Please give a big hand to Professor Dr. Shahid again. Thank you so much, sir. Now there is the poster session and tea will be served there. There is a competition. We have 3 certificates for the first 3 positions. So all the poster participants are requested to please go to the venue. Jury will come there and analyze their posters. We will start the next session at 4:30 after 30 minutes session. Thank you so much. May I request the speakers of next session to please copy their presentations. We are here for this purpose.
Enjoyed your talk? It's good. So how is the situation? Sure, I was going to go to this gentleman or wait. Okay, thank you a lot. Now is tea break.
去喝点咖啡去。
नहीं हमने आपसे तो वैसे ही किया हमने तो इंतजार से किया पता नहीं कैसा शो करते हैं
Well, so there's not short answers. The short answer.
मैडम इसको क्या प्रॉब्लम थी?
Dr. Rehan from Food and Mobility Research Center. Our dean, she's the head of the R&D at FMDRC. Nice to meet you. She's also in FMDRC. She's also looking at it.
Nice to meet you. Nice to meet you.
Nice to meet you also.
Yeah.
I have already meeted you.
Yes.
मैं आपको दे देता हूँ इनका है। वो जो आपने मुझे भेजी है।
While sequencing of our old serotypes.
Yes.
Maybe you remember
Probably,
make
yeah.
a phylogenetic tree for us.
Yes.
Well, you was the most concerned person for us.
We have displayed that phylogenetic tree.
Okay.
Okay. Our main purpose to come out here just to listen to you.
Okay.
You're also from the family and you're also
now you're flattering me. Yeah.
No,
Research paper you we give your name.
Yeah, okay. He's also working with
Nice
to meet you.
also
in
Nice to meet you.
FMDRC.
We are the team.
Okay. So you're the national reference body for FMD?
We have the only one research institute on FMDRC in Lahore.
Now
We are
we
working
have
with them.
national reference body status now.
Good. Yeah, the space is very confusing for me. Lots of institutes for that.
But you can't forget FMD.
Yeah,
because
no,
you're also the expert of
yeah.
FMD.
And we work a lot, of course, with the samples that come from Pakistan, with the FAO office.
Yes.
Yes, definitely we can't deal directly. We have to deal through the FAO.
And I heard Dr. Afzal is retired.
Yes, Dr. Afzal has retired.
Who will be the new person?
Dr.
Akram.
Dr. Akram.
Could you
but he's
get
not,
his email for me?
Sure, I can send you.
Yeah.
I have your email also.
Yeah, that would be really helpful because Dr. Afzal has been my big point of contact for many years. And
Tell me your email.
I can give you my card.
He's not right now permanent.
Okay.
Because I think they're waiting for the permanent.
Yeah.
Okay, thank you. Well, I will share with you.
Yeah, that would be really helpful to get his replacement. I wrote to him about 2 weeks ago, and he said that he is no longer working for the FAO. He's now working for, I think, a university. He was giving me the impression he was still doing some work.
But we can visit
Nobody
our
went.
FMDRC.
Okay.
Because he always represented all the government institutes at the international level.
Yeah.
It's nice, very nice to meet you.
Yes, very nice to meet you.
Comfortable.
While we, I think we'll be staying that new institute.
Okay. I have my flight tomorrow, unfortunately.
Okay, you're going back.
Yeah.
We wish you can visit.
Yeah, I would love to.
FMDRC.
You have to visit.
Next time I come, I think.
Which was the expert of the family who visited for the SOPs and
Danny
Hoover. Jennifer.
Danny, no, Dr. He was Danny from Brazil, I think. Danny from Brazil.
He spent almost 1 to 2 weeks at FMDRC. 15 days at FMDRC.
Danny.
So Danny.
Okay. Well, if you need anything, just you have my email now. So
Sure.
no excuses. So just send me an email, and I can. Yeah. Good. Nice to meet you. Sorry.
मैडम जी और कितना हमारे लाए
तो बिस्किट ला दें केक ला दें
अल्लाह
और क्या करूं करो सत्य कह रहे थे
मैडम जी वो चाहे शुरू आ रहे
वही जा रहे हैं
लेकिन अलीना ठीक है पीछे करवाया हमने
और पीछे मुझसे तो करा रखते हैं जरा तो मैं बैठे हैं
फिर वो उसको तो बिल्कुल नजर नहीं आएगा ऐसा नहीं
हम इसको दो को एक को निकाल के एक टेबल पर रख देते हैं तो साइड पे कर देते हैं छोटी चेयर रखवा के ना उससे वही टेबल बड़ा रहने दें ये पूरा हट जाएगा तो उन दोनों को पास नजर आएगा चेयर छोटी कर देते हैं
मजा नहीं आएगा छोटी चेयर
हां नहीं तो ये कुर्सियां थोड़ी इधर ही कर दें वैसे
बड़ी कर देते
हां ऐसे कर दो
हां जी इधर कर दें अरे मैं
ये इधर कर दें वो टेबल आगे ही लगा दें वो टेबल ना हटा दें
या टेबल से नीचे कर लेते हैं मैडम हां ये तो भीड़ पे तरफ फेंक लेते हैं
पहले वो टेबल हटाएं हम समझ में आती है वो टेबल से नीचे आ जाएगी उसकी चीजें कहां शिफ्ट करें उधर शिफ्ट कर देंगे
भैया टेबल बैठे थे
नहीं मैडम वो स्ट्रेट हो जाएगा सीन जस्ट डबल नो
अच्छा हां ये इसकी वजह से कर रहा हूं तो इसको फिर
ये इसके नीचे लग जाएगा जैसे वो
इधर कर दें
हां
ठीक है
बोतल मेरे देख लेना
ये ले
यहां से
अल्लाह लो
ये चीज साथ साथ करें
एक फाइल मैडम जी आज मैं ड्यूटी कैसी की है सर? अच्छा ऐसे किसी ने बताया मैडम आपने
मैडम खाली है मैं हम 4 लोग हैं तो दिए में इत्तेफाक में और मैडम बनी
जी अस्सलाम वालेकुम सर।
आज मैडम हम यहां बहुत से फैक्टर तैयार है
जी जी सर। नहीं सर कोई तैयारी नहीं रह गई जी साफ सुथरा कमरा है
खत्म हो गया कहां से मांग रहे हैं
कौन हो गए नहीं ਸਾਨੂੰ ਕੋਈ ਨਜ਼ਰ ਨਹੀਂ ਸਰ ਦੋ ਪਹਿਲਾਂ ਸੀ ਤੇ ਉਹ ਅਸੀਂ ਬਾਅਦ ਜਦ ਬੰਦੇ ਨੂੰ ਨਿਕਲੇ ਅਸੀਂ ਦੱਸਿਆ ਤਾਂ ਉਹ ਜਿਹੜੇ ਸੀ ਡਾਕਟਰ ਦੇ ਦੋਸਤ ਆਏ ਸੀ ਉਹਨਾਂ ਨੇ ਸੀ ਉਹਨਾਂ ਸਾਨੂੰ ਲੈ ਲਿਆ
उन्होंने मुझे कहा कि पानी ढूंढ के मैं इधर से
ਆ ਸਰ ਪਹਿਲਾਂ ਨਹੀਂ ਆਇਆ ਅਸੀਂ ਨਹੀਂ ਦਿੱਤੇ ਉਸੇ ਕੋਲ ਨੂੰ ਵਸੇ ਉਹ ਸੀ ਪਹਿਲਾਂ ਪੈਨ ਕੋਲ ਵਿੱਚ ਤੇ ਪਹਿਲਾਂ ਨਹੀਂ
खुली नहीं है सारे
ਹਾਂ ਜੀ
ये वो ग्लास भी
ਸਾਰੇ ਤੇ ਦੇ जी जी वो ऐसा बेबाल डॉक्टर गेस्ट नहीं आए एक तो मैडम आए थे ना पर एक चैनल मैडम है बड़े सारे मैडम है उन्होंने ये ठीक है ये साइड पे करते ते एक जना डॉक्टर साहब आए ने यार ना उन्होंने फाइल उन्होंने खुद ही फाइल फड़ी आप अरे मेरी फाइल मेरी जगह से
डॉक्टर नूरीन एंड डॉक्टर समरा
नूरी भेजे मैडम समरा
शहजाद और डॉक्टर नूरी इत्तेफाक पानी की बोतलें हैं
ये है मैडम कितनी कितनी चाहिए
3-4
3 तो 4 7 होते हैं
6 दे दो भाई
एक काम
ना पीना पड़ी
आज ठीक है नीचे से बैठ के देख सकते हैं कैसे लग रहे हैं बैठे हैं
नहीं ठीक हो गया फर्स्ट क्लास वाला सिर्फ 2 लोग हैं
अभी 2 होंगे सुबह में 2 होंगे
ये एक दफा ना जी इनको चाहिए ये देखना डॉक्टर साहब
हाँ जी
ये चीजें छोटी छोटी ये उठा लें यहाँ से
किधर है यहाँ
बोतलें